Hi, everyone. Welcome to this video lecture on enzymes. So enzymes are actually a subset of the protein family of macromolecules, but they are important enough that they get their own video dedicated just to themselves. So we'll start by talking about what enzymes are and what is it that they do. So essentially, enzymes are proteins that speed up or catalyze chemical reactions. A couple special properties about the way they work. They're not actually destroyed by the reaction. So you can reuse enzymes for the same reaction many, many, many times over. So it's not destroyed by the reaction that it's catalyzing. And each enzyme is specific to a particular reaction. So you have hundreds of different enzymes in your body that each are specializing in a particular reaction that they'll speed up. Um, Enzymes are especially common and necessary for metabolism. And just as a reminder, metabolism is referring to all the chemical reactions that are taking place within an organism. So, pretty much any metabolic reaction needs an enzyme in order to happen at a reasonable rate. Otherwise, it would be so slow that the organism would die. So, before we get too far into this, I just want to introduce a couple of vocabulary words. So, essentially, what enzymes are doing are they're taking molecules called substrates and changing them into different kinds of molecules called products. So, in short, we can say that enzymes are taking substrates and turning them into products. So, the substrates are molecules that will undergo a reaction once they bind to an enzyme. And the product is the molecule that's the result of the reaction. And then on the enzyme itself, it has this special space called an active site. And this is the particular spot on the enzyme where the substrate actually binds. So here you can see the active site is this sort of groove here with this other groove here, and that's where the substrate fits in. Once the substrate binds, the shape will change a little bit as it's to sort of fit the shape of the substrate. And then the enzyme will transform that substrate into the product, and then the product will leave and dissociate from the enzyme. So the active site on the enzyme, its shape and its chemistry are going to be especially fitted for the substrate that it's supposed to be transforming. And then the enzyme will encourage bonds to be broken or formed within that substrate in order to transform it into the product. So we're going to start by looking at a familiar example, which is lactase. So lactase is responsible for breaking down the sugar lactose, and that's found in a lot of dairy products. So lactose is a disaccharide, which means it's made up of two monosaccharides, and specifically those two monosaccharides that lactose is composed of are glucose and galactose. So, in this case, we would say that the substrate for the enzyme lactase is the lactose molecule. That's the thing that's going to undergo a chemical reaction. And the products are glucose and galactose, those two separate monosaccharides. And we can also see that this lactase enzyme has a special active site that is specifically shaped for the lactose substrate. So if a different disaccharide were to come in, say sucrose, it wouldn't fit into the active site for lactase and nothing would happen. So that's one example. And then a fun fact is that most mammals only make the enzyme lactase during infancy when they're actually drinking milk. So it wasn't until humans started domesticating cows that the ability to digest lactose past infancy became a more common trait. So in a lot of places around the world, lactose intolerance is actually more common than being able to tolerate lactose. Okay, so this is just a separating two different kinds of metabolic reactions. We said metabolic reactions are all the collection of chemical reactions that happen within an organism, and there are two main types. So synthesis reactions are also said to be anabolic, and synthesis reactions are ones that are building things up, so creating larger molecules out of smaller molecules. So you can see an example here. Two substrate molecules are being joined together to make a larger product. And then the opposite is decomposition reactions. So these are said to be catabolic instead. And this is where we're taking a larger molecule and breaking it down into smaller pieces. So in this case, the substrate is two things that are already joined together. And then when it goes into the enzyme breaks the two things apart, and we end up with separate products. So this would be more similar to the lactase example that we just looked at. Okay, so when we're thinking about how quickly an enzyme catalyzed reaction is able to occur, there are a couple things for us to think about. So the rate of the reaction, or how quickly it proceeds, is determined by two main things. One thing is how often the enzyme and the substrate collide. So in order for a reaction to occur, the substrate and the enzyme have to actually like bump into each other. They have to come into contact. So, how often that is able to happen is one determining factor. And then the other determining factor is whether the enzyme and the substrate can actually fit together properly. So, one thing that can happen is that since enzymes are proteins, they can also be denatured like proteins. And remember that when a protein is denatured, it's going to lose its shape. And in the case of an enzyme, if it loses the shape of its active site, then the substrate might no longer be able to fit into that active site, and then no reaction will happen. So, we're going to take a look at some major factors that affect reaction rates and talk about them one by one. So, we'll talk about regulators, we'll talk about concentrations, and then we'll talk about external conditions. So we'll start with this slide on regulators. Regulators can actually encourage a reaction to go forward or actually slow it down as well. So there are two different kinds, and they'll do two opposite things. So inhibitors are a kind of regulator, and as the name suggests, they're going to reduce the reaction rate. So they're going to prevent a proper interaction between the enzyme and its substrate. And then on the opposite hand, we also have activators that can increase the reaction rate. So activators somehow encourage the proper interaction between the enzyme and its substrate. There are two different kinds of ways that these inhibitors can work. So we've got one more slide focusing in just on these inhibitors that are going to reduce the rate of a reaction. So again, inhibitors are going to prevent an enzyme from working properly, and there are two places that an enzyme inhibitor can bind. So sometimes the inhibitors will actually bind the active site. And so you can probably easily imagine how if the active site is directly being blocked by a different molecule, then the substrate can't get into that active site, and then the reaction will not be able to occur. So when an enzyme binds at the active site, we call that competitive inhibition because the inhibitor and the substrate are competing for the active site. And then inhibitors can also bind somewhere else besides the active site. And usually, when they bind somewhere else, they're going to change the shape of the active site or the rest of the enzyme so that the substrate is unable to bind. And so, when an inhibitor binds to somewhere other than the active site, we call that non competitive or allosteric inhibition. So, those are diagrammed right here. Competitive inhibition is when the inhibitor is binding at the active site and then substrate is blocked from binding. Non-competitive or allosteric inhibition is when the inhibitor binds somewhere else, but it still changes the shape of the active site so that the substrate is unable to fit in. Okay, so here on this slide, we're talking about concentrations, and this can refer to concentration of either the enzyme or the substrate. And in general, here, having a higher concentration of the enzyme or the substrate will increase the reaction rate. And this is because the more enzyme or the more substrate you have, the more likely it is that the two are going to bump into each other, and you'll get that reaction to go forward. So remember that we said that you need to actually have the enzyme and the substrate colliding with one another and making contact in order for the reaction to proceed. So that's more likely if we have a higher concentration of enzyme or a higher concentration of substrate. There is a point that we call saturation, where essentially all of the active sites on the enzyme are occupied. So even if we keep increasing the concentration of substrate, there's just no more spaces for them to be converted into product. There's no more active spaces available, active sites available. So we basically just have to wait until the reaction occurs, and then that enzyme will be freed up, and then the substrate can take a spot. So the saturation point is when all the active sites are taken up, and we're kind of at our max rate for the enzyme. Okay, and then these last two slides have to do with external conditions. So the first one that we'll discuss is temperature. And for temperature in general, higher temperatures is going to increase the reaction rate. And that's because as there's more heat, so a higher temperature means there's more heat, it means that the molecules are going to be moving around faster. And if they're moving around faster, then they're going to be colliding more often. I think you could imagine this like in the case of a bunch of people. If you're walking around in a room very slowly, you're probably not going to be bumping into anyone. But if you have just everyone sprinting as fast as they can around a room, it's more likely that two people are going to collide, right? So, same thing with enzymes and their substrates. If they're moving around more, if there's a higher temperature, then there are going to be more collisions, so a faster reaction rate. So increasing temperature tends to increase reaction rate, and generally each enzyme is going to have an optimal temperature where its reaction rate is the highest. So for humans, it'll tend to be around our body temperature, which is about 37 degrees Celsius. But that could be different for different organisms that live in different temperatures. So the optimal temperature for an enzyme for a hot spring bacteria might be a lot higher. However, I did say in the protein video lecture that proteins will denature at really high temperatures. So we use the example of cooking an egg and seeing the egg yolk turn from translucent to white. So since enzymes are proteins, they also will denature if the temperature gets too high. And in that case, they're going to lose their shape and reaction rate is going to go down to zero, essentially. So there's an optimal rate, but if it goes past that, then we'll denature our enzyme and the activity of that enzyme will go away. And then finally, pH works in a similar way. Each enzyme has an optimal pH where its reaction rate is the highest. So here, pepsin is an enzyme that works in our stomach. So you can see it has an optimal temperature at a really low or acidic pH. And salivary amylase is as the name suggests, is an enzyme that is found in our saliva, so a sort of a neutral pH. Um, but if the pH gets too far outside of that optimal range, then the enzyme will denature, and again, the reaction rate will decrease. So both temperature and pH are external factors that can influence the reaction rate of an enzyme. Okay, so that's our enzyme video lecture, and I will see you all soon.